Welcome back, everyone. And so we're going to be finishing up our notes for this week on membrane transport and facilitated diffusion. And so we're going to just jump right into it. And so we're going to talk about the selective permeability of the plasma membrane and what that means. And so what I mean by that is that some substances can easily cross the membrane than others. Things that have an easy time are going to include small nonpolar molecules like CO2, oxygen, and nitrogen, and hydrophobic molecules as well. Hydrophobic molecules. What's going to have a hard time getting through the phospholipid bilayer are hydrophilic molecules. Polar molecules and large molecules and ions. And so things like sugar, water, sucrose, chlorine ions, and sodium ions, all those things. And so we see that these small nonpolar molecules have a good time, these uncharged molecules have an easy time, whereas large molecules, they have a harder time, and so do these ions. However, these molecules are still able to get through the phospholipid bilayer, but we'll talk about how they do that in just a moment. And so we have two different types of transport. And so hopefully this is a little bit of a review. We have two types of transports that happen on the plasma membrane. We have passive transport and active transport. And so let's just talk about passive transport first. Passive transport is the transport of a molecule where it does not require energy, no ATP required from the cell, because we are moving from a concentration gradient of going from high to low. A high concentration to a low concentration. No energy required. And so some types of passive transport are going to include diffusion, osmosis, and facilitated diffusion. And so we should have already have taken these notes, and so I'm not really going to elaborate on this too much. I'm just going to show you the screen. Diffusion is the movement of molecules from areas of high to low. So I'm going to highlight just the key vocab. And we can see this happening in the image below, the sugar molecule dissolving across the whole water substance evenly. We have osmosis, which is just diffusion with water. And so now we're in our new part, the part that we don't have notes on, which is facilitated diffusion. Facilitated diffusion is the diffusion of molecules through the membrane via transport proteins. And these transport proteins are going to help with increasing the rate of diffusion for water, small ions, and carbs. And so we have two types. We have channel proteins. Channel proteins provide a channel for molecules and ions to pass through. And so basically they are a particle-specific door that opens up for certain molecules. And so one example is going to include aquaporins. This specifically allows water to come in and out of the cell, or just in or just out of the cell. And these channels can open and close depending on what the cell needs, and they will open and close them with signals. So we have channel proteins, which are just open channels for things to come in. And then we'll have carrier proteins. And so what will happen is that a carrier protein will undergo changes for substances to pass. And so what we'll see is that we'll have this open structure right here, this Pac-Man shape right here. We're going to get an ion that's going to fit into a special place inside this Pac-Man's mouth. And what we'll see is a change in the shape. And we'll see what we'll see is that the door is actually going to switch. It's going to invert itself, and it's going to open up and release this molecule to the inside environment. And so we see it changing shape, changes shape to let substances pass. And so let's think about it. What's faster or slower? A channel protein or a carrier protein? Channel, because things just need to flow into it, whereas the carrier protein has to wait for a molecule to come into the carrier protein. We have to wait for it to change shape and then wait for it to release that said molecule back into the inside of the cell or the outside. Facilitated diffusion is still passive transport just because the proteins are moving stuff from an area of high concentration to low, which requires no energy. And so the types of passive transport will include facilitated diffusion via carrier protein or channel protein, or via osmosis or diffusion. And so we finished talking about passive transport. Now let's talk about active transport. Active transport is the opposite. It's going to require energy. It requires energy. And we're going to be moving against the concentration gradient. And so we're trying to move from concentrations of low to concentrations of high. We're going against the flow. We're going against the stream, the current. And we have a couple types of active transport that we'll talk about. We'll have pumps. Think of like a pump to like get water into a fish tank. We'll have co-transport. We'll have exocytosis and endocytosis. And we'll go over all these in just a moment. And so first off, what is energy? The currency of energy that our cells use is ATP. Adeno triphosphate, which is a nucleic acid. And we use ATP as a source of energy, an energy source for cells. And we use ATP on our day-to-day basis. And how ATP is going to work is that we're going to have energy. We have to release a phosphate group. We're going to have energy stored in these bonds right here. And these bonds that connect the phosphates to the nucleotide. And when we break these bonds, when we break a bond, we're going to release the phosphate. It's going to release a phosphate, and it's also going to release energy. And so we're going to first talk about pumps. Pumps maintain membrane potential. And what that is an intentional, very intentional, creating an unequal concentration of ions that will cause an electrical charge. And so what we see in here is that we have a ton of sodium ions, these purple guys right here. We have a ton of these in here. They're all positive. And they are going to want to move to the outside. They want to move on the outside, but we are keeping them in on the inside. We are also going to have a bunch of chlorine ions, these green guys. And then we'll have potassium ions on the outside. And these help with neurons and electrical currents and allowing for messages to transport between cells at a rapid rate. So some examples of pumps are going to include electrogenic pumps, and these are going to generate voltage. They're going to create electricity across our membranes, which we then use as an energy source for cellular processes. One example of this electrogenic pump is going to be the sodium-potassium pump, probably one of the most important things for this unit, the sodium-potassium pump. It only exists in animal cells, and it regulates the concentrations of sodium ions and potassium ions in the cell. What's going to happen is that the cell is going to absorb or bring in 3 sodium ions into it. Sorry, pump out 3 sodium ions and pump in 2 potassium ions, which will cause a positive charge on to the extracellular fluid. And so here's an image that kind of describes this better. So think back to facilitated diffusion. We're gonna have not a channel, oh my gosh, I forgot the name of it. Let me zoom back real quick. Carrier? I'm pretty sure it's carrier. Yeah, carrier. Think of these carrier proteins. Think of the carrier proteins in facilitated diffusion. This structure is gonna wait. This structure is going to wait for sodium ions to go sit. 3 sodium ions are going to sit inside its slots. And what's going to happen is that the sodium potassium pump will change shape. It will first use energy. It's going to use some energy. It's going to change shape and release these sodium ions. And it's going to leave this newly open protein for potassium to go fit in its special seat. And then the protein will then fold again or change shape again, releasing the potassiums back into the cell, creating a more positive environment on the outside. We start off with negative on the inside and positive on the outside. Let's flip that. Positive on the inside, negative on the outside. And after all these reactions happen, we flip that. And this will create an electrical current, which allows for cells like neurons to transmit messages at a rapid rate. Okay, so some other types of active transport are going to be exocytosis and endocytosis. Exo is going to represent out, to secrete. Exo is going to be out, whereas endo will be in. And so what exocytosis is going to do is that exocytosis is going to secrete molecules via the vesicles from the Golgi apparatus. And so what we're going to see is that we're on the inside of the cell over here. We're on the inside. We're going to create a little vesicle, and we're going to take nutrients or molecules or whatever we want, and we're going to spit them back out into the environment. Whereas with endocytosis, it's going to be the opposite. It's going to be the uptake. And so let's say we have sugar floating around. The plasma membrane will detect that the sugar is on the boundary, the outside boundary of the plasma membrane. It's going to engulf it and bring it to the inside of the cell. And this is how we get big molecules in, like glucose. And so it's the uptake of molecules and materials via vesicles into the cell. And we'll have 3 types of endocytosis. The first one is going to be phagocytosis. And this is going to be think of eating. This is cell eating. And this is when the cell engulfs a particle that we want to digest for later in the lysosome. And so they will package this material, this particle, into a food vacuole, and that food vacuole will eventually find its way into the lysosome and get digested. Our next type of active transport is going to be penocytosis. Think of cell drinking. And so what penocytosis is going to do is that it's going to take in a bunch of material from the outside environment, engulf it into a vesicle, and bring it to the inside. And then those molecules will eventually get released into the extracellular fluid on the inside of the cell. And these could be hormones, they could be messages, they could be other ribosomes, they could be a variety of things. And then our last type of active transport that we'll talk about is receptor mediated endocytosis. It is a specific uptake of molecules via a solute binding to the receptors on the plasma membrane. A lot of words. So what this says is that there is these special particles right here that are on the lookout for, let's say, these particles really want some sulfur ions for some reason. They want sulfur. And so these are going to be sulfur specific. And they will wait until sulfur is attaching to these how do I phrase this? These receptors. And once they have collected enough sulfur, it will then engulf it into a vesicle and bring it to the inside. And so this allows for the cell to take up large quantities of a certain substance, like sulfur, maybe iron, maybe a neurotransmitter, maybe a hormone, and they get brought into this they get brought to the inside of the cell thanks to a specific receptor, receptor that allows for these substances to be detected, attracted, and brought into the cell. And so that is the end of these notes, and we will continue taking notes for the rest of this unit in class together.